All right, I think we're going to get started here. So welcome to the next installment of the QBI-GIV Infectious Diseases and Human Health Seminar Series. And we're delighted to have Jesse Bloom here from the Hutch. He's a professor at the Hutch in the Division of Basic Sciences and Computational Biology. He's also a professor at the University of Washington. He's been an investigator of HHMI since 2018. He has a whole slew of awards and honors, just to name a few. My favorites: the Searle Scholar, Pew Scholar, and Burroughs Wellcome Investigator in the pathogenesis of infectious diseases. And as a lot of us know, Jesse's really a world's leader in studying viral evolution using a variety of different approaches. And he's having a really big impact in studying the evolution of SARS-CoV-2. He's been doing some really elegant work on the systematic mutagenesis of different regions of SARS-CoV-2, including a focus on the spike protein. And that's giving us a lot of insight into ultimately the immune response. And I'm very excited to be here, what he's going to talk to us about with respect to SARS-CoV-2. I think he's going to talk about SARS-CoV-2, and his talk is entitled Interpreting the Evolution of SARS-CoV-2. So Jesse, we look forward to your talk, and you can take it away.
Okay, thank you for the kind invitation. And I just posted the link to my slides in the chat. So hopefully that went to the audience as well as the attendees here. If not, I apologize, and it's on my slides, so you can email me the slide link. Okay, so I'm going to talk today about interpreting the evolution of SARS-CoV-2. So as I just mentioned, the link to the slides is right here and is hopefully in the chat. And you're welcome to kind of follow along because there's a few interactive things if you want to look during the presentation. Okay, so since I'm going to be talking about viral evolution, I'd like to start by taking a step back and pointing out that for most of the history of evolutionary biology, people didn't know anything about DNA or RNA or genes or genomes. So here's Ernst Haeckel's Pedigree of Man, which is this famous image in the history of evolutionary biology. And in this pedigree, he's arranged these different species in this tree-like structure based simply on looking at their phenotypes and kind of putting the ones that look similar to each other close by in the tree. So it was only about 55 years ago that it became possible to also start to study evolution in a different way, which was in terms of changes in genotype. So one of the first papers of this type was by Emil Zuckerkandl and the famous chemist Linus Pauling. And in this study, they'd been able to obtain the myoglobin sequences from a variety of different species. And they arranged these myoglobin sequences in a tree-like structure so that the ones with similar sequences were sort of closer together in the tree. And actually at the time, this idea of studying evolution this way was really intensely criticized by a lot of famous evolutionary biologists like Ernst Mayr or George Gaylord Simpson as being really trivial, because their argument was, what could you hope to learn about sort of the important characteristics of horses and humans and chickens and so on, just by looking at the sequences of these myoglobin proteins. And in fact, Zuckerkandl and Pauling recognized this criticism, and they noted that how changes in the genotype of these proteins affect their phenotype would be complicated. They wrote, there's no reason to expect that the extent of functional change in a protein is proportional to the number of amino acid substitutions. They said it's the type rather than the number of amino acid substitutions that would be decisive. So now, obviously, 55 years later, we've gotten really good at characterizing the evolution of viruses and viral proteins at the level of genotype. So for instance, you can now go to public databases and download millions of full-length SARS-CoV-2 genomes, and you can build these ridiculously high-resolution phylogenetic trees that tell you in great detail how the virus is changing at the level of genotype. However, the question of interest is still often actually how these changes in viral genotype are going to affect the phenotype. So here's an article on this topic called "The Coronavirus is Mutating. What does that mean for us?" And the point I just want to make in this introduction is that this question that a lot of people have been asking over the last couple of years is really just a special case of this broader question that evolutionary biologists have been thinking about for a long time, which is, what is the relationship between sort of the number of mutations you might observe in a protein, which is now quite easy to determine, and the type of functional change those mutations cause, which might be much harder to figure out. And so my lab is interested in studying this question of how changes in the genotype of a protein relate to its phenotype. And we're interested in doing that in the context of viral proteins. So to be very specific, we might want to have some viral protein and understand if you make some mutations to it, how is that going to change biochemical phenotypes that we think are important for the function or the fitness of the virus out there in nature. So today I'm going to talk about studying this mapping from changes in genotype to phenotype in the context of SARS-CoV-2. So the outline of my talk is going to be broken into the following 4 parts. First, I'm just going to give some brief background on the SARS-CoV-2 spike and its receptor binding domain. Then I want to talk at sort of a general level about how coronavirus spikes evolve in terms of changes in genotype and phenotype. Then I want to zoom in on this question of how mutations affect the receptor binding domain of SARS-CoV-2, and finally, how these mutations might affect the ability of antibodies to recognize this part of the virus. Okay, so probably most people know this, but just to start with a little background. On the surface of SARS-CoV-2 virions are many copies of this protein called spike. So this is a large class 1 viral fusion protein, and the function of spike is to get the virions into the cell. And so one key part of the spike is the receptor binding domain, which is an autonomously folding subdomain of the spike that I've colored here in purple. And the function of the receptor binding domain is to bind with high affinity to a protein called ACE2, which is found on the surface of some human cells. And so this high affinity binding of the receptor binding domain to ACE2 is the first step in the initiation of cellular infection. And we also now know, and I'll go into a little more detail on this later in the talk, that neutralizing antibodies to SARS-CoV-2 invariably target the spike, and they often target the receptor binding domain, or RBD, and in some cases act simply by blocking its interaction with ACE2. And so for all these reasons, understanding both the evolution of spike and in particular its receptor binding domain is quite important. Okay, so that's a little sort of high-level background on spike. Now I'd like to ask the question, sort of, how do coronavirus spikes evolve? Is the evolution of this protein going to be important long-term? So hopefully most of you know that only sometimes does the evolution of a virus lead to changes in its actual antigenic phenotype. So 2 classic examples of this are 2 different respiratory RNA viruses. So these are 2 different human viruses. They're both RNA viruses. So they both have very high mutation rates, and they both transmit by the respiratory route in humans. However, measles virus does not evolve to escape immunity. So in the old days, people were infected at most once in their lives, and then a vaccine was developed in the 1960s, and that vaccine still works very well today. In contrast, influenza virus is another respiratory RNA virus that does evolve to escape immunity. So the typical person is infected with any given subtype of influenza virus about every 5 years, and the vaccine needs to be updated annually because the virus is constantly changing in a way that's eroding antibody immunity from prior infections. So an important question is, where might SARS-CoV-2 fall if we sort of define the spectrum from measles virus to influenza virus? Because SARS-CoV-2 is obviously a 3rd respiratory RNA virus. So early on in the pandemic, many people suggested that coronaviruses like SARS-CoV-2 would not evolve antigenically. So for instance, here's an article on this topic from early 2020 from the Washington Post entitled, "The Coronavirus Isn't Mutating, Suggesting a Vaccine Would Offer Lasting Protection." And the argument that was given to support this notion is sort of as follows. Coronaviruses are the only RNA viruses with a proofreading polymerase. And that proofreading polymerase means that they have a somewhat lower mutation rate than other RNA viruses like influenza virus or measles virus. However, I'd still like to point out that the mutation rate of coronaviruses is still appreciable. It's just not quite as high as for influenza or measles. In any case, we didn't think that this argument here was very compelling. And the reason we thought it probably wasn't a good argument is as follows. Evolution isn't the same as just mutation. Rather, evolution involves 3 related factors. So one of those factors is indeed the underlying rate at which mutations arise. But after the mutations have arisen, what happens next depends on both the phenotypic impact of those mutations and also how natural selection or genetic drift act on those mutations. And so we felt like we really needed to understand these other two factors as well to try to understand how a virus like SARS-CoV-2 might evolve. So given that those are really complicated to understand, we kind of tried to take an indirect approach, which is just based on the idea that the best predictor of future behavior is going to be past behavior. So there's no extensive past behavior of SARS-CoV-2 itself to study, but we can study the relatives of this virus. So as probably most of you are familiar, SARS-CoV-2 is not the first coronavirus to infect humans. There's actually several endemic, so-called common cold human coronaviruses that are thought to cause about a third of all common colds. And one of these is called coronavirus 229E. And this is a virus that's been circulating in humans since at least the 1960s when it was first described, and probably well before the 1960s. So coronavirus 229E and these other common cold coronaviruses hadn't been extensively studied because they typically did not cause severe disease, but they've certainly been evolving in humans for quite a while. And so we wanted to see, is there evidence of antigenic change during the evolution of coronavirus 229E? So what we first did is we just went to GeneBank and we built a phylogenetic tree of all of the coronavirus 229E spikes. So in this tree, the oldest spikes are back here from the late 1970s and early 1980s. And then down here on the lower right are sort of modern sequences from the last few years. And so there's a few things I hope you can appreciate about this tree. One is that this tree has what's often referred to as a ladder-like topology. So there's a major trunk lineage here that I'm tracing along with my mouse, and then a lot of short side branches coming off that trunk. And this is indicative of a virus that's changing through time. So there's this major trunk lineage, but one variant or lineage is constantly sort of winning out and displacing the others. So all of the side branches are short and sort of die out. So this is what a phylogenetic tree of, for instance, human H3N2 influenza would look like. There's a lot of change over time, but the standing diversity at any given time is relatively low. Importantly, this is different than what a phylogenetic tree of something like HIV would look at. Whereas HIV, there's a lot of long coexisting branches because the virus has high diversity at any given time as well as over time. So one feature of phylogenetic trees with this shape is that sort of where you are on the x-axis, which is the time axis, is a pretty good indication of where you are overall in the evolution. So what we did is we just used these data to experimentally generate coronavirus 229E spikes at 8-year intervals along the tree, 1994, 1992, so on. And then we used these to generate pseudotype lentiviral particles that we could study in the lab. So we then did just a super simple experiment. So we took old human serum samples. So on this slide, I'm showing a human serum sample from 1985. And then we asked, how well can that old serum sample neutralize both sort of the contemporaneous viruses? So for instance, this virus from 1984, and this person might have been exposed to a similar virus, and also viruses that are sort of in the future from 1984. So 1992, 2001, so on. And so the x-axis here is just the year that the spike come from, and the y-axis is the neutralization titer, which is the reciprocal serum dilution needed to neutralize 50% of the virus. And so for comparison, if you've received a single series of SARS-CoV-2 vaccines, or if you've been infected with SARS-CoV-2, you'll typically have a neutralization titer that's, on the order of 500 or so. So this individual can neutralize the coronavirus 229E from 1984, almost as well as someone who's gotten a SARS-CoV-2 vaccine could neutralize SARS-CoV-2. But then as that virus evolves forward in time, its evolution of the spike is eroding this antibody neutralization. such as by 2008, the virus has changed so much that this serum no longer works against it. And importantly, I want to emphasize this plot is not showing antibody durability. So antibody durability is another important immune parameter that many people are characterizing. And antibody durability is essentially if you take serial serum samples from the same person, so you collect the serum samples over time, how does their ability to neutralize a single virus change? And so that's important because it tells us how the potency of our antibody-mediated immunity might change over time. But this isn't showing that. It's the same serum sample for all these points. And so all of these changes here are due solely to changes in the virus and nothing to do with the human individual. And we've looked at other individuals. You can look at a lot of these plots in this paper. I'm just showing a few more here. And one of the consistent things you can see is that the viral evolution always erodes the neutralizing antibody titer. So in some cases, like this individual here, this erosion is extremely fast. So the neutralization titer is undetectable after 8 years of viral evolution. Here's a sort of gradual evolution I was showing on the previous slide. And then this individual's neutralizing antibodies are being eroded by viral evolution, but only very slowly. Even 30 years later, they still have appreciable neutralizing titers against the virus. And so one of the questions that we're currently researching in our lab is what are the differences in the antibody specificity between this type of evolution-resistant antibody immunity to coronavirus 229E and this type of evolution-sensitive neutralizing antibody immunity. And the goal of understanding that, of course, is if you're making a vaccine against a coronavirus, ideally it would elicit neutralizing antibodies that are pretty resistant to viral evolution. So it'd be nice to know where naturally evolution-resistant immunity is targeting on the virus. And then the last point I just want to make, which will be of most interest to any of you who are sort of real SARS-CoV-2 aficionados, is where the mutations are occurring in the spike. So the coronavirus 229E spike is broadly similar to the SARS-CoV-2 spike. It has an S1 domain and an S2 domain. And then the S1 domain has two major subdomains, the N-terminal domain and the receptor-binding domain. So the actual receptor for coronavirus 229E is aminopeptidase N. So it's a different protein than ACE2, but the basic structural principles are the same. And so down here, I'm plotting where over that time frame from 1984 to the present are mutations occurring in coronavirus 229E. And you can say that the largest number of mutations are in the receptor binding domain, particularly in sites that are contacting or near those that are contacting the receptor. And then there's also quite a few mutations in the N-terminal domain and fewer mutations elsewhere. And so for those of you who follow SARS-CoV-2 variants of concern, you will notice that this is essentially exactly where mutations occur in the spikes of SARS-CoV-2 variants of concern. Essentially, every SARS-CoV-2 variant of concern has mutations in the N-terminal domain and mutations in the receptor binding domain, often in ACE2 contact sites. So it looks very, very similar between this virus and what we see for SARS-CoV-2. The main difference in the patterns in SARS-CoV-2 is there's also an enrichment of mutations in SARS-CoV-2 variants of concern in the furin cleavage site in the S1, S2 boundary region. And that's probably simply because SARS-CoV-2 is not currently well adapted to its furin cleavage site and is still sort of adapting there, whereas coronavirus 229E is clearly well adapted to the furin cleavage site it has, or the protease cleavage site it has. Okay, so now I've talked a little bit about the general principles of how coronavirus spikes evolve. Now I'd like to talk about the question of how mutations specifically affect the function of the SARS-CoV-2 receptor binding domain. And the reason we care about this is to say something useful about SARS-CoV-2. We not only have to know that there might be antigenic evolution, but we actually want to know how mutations are directly affecting phenotypes like antigenic recognition. So our approach is to sort of prospectively measure for how this receptor binding domain, how each mutation affects key phenotypes. And the reason I use the term phenotypes is, of course, what we would really like to know is how these mutations are affecting viral fitness. However, the fitness of a human respiratory virus like SARS-CoV-2 is a property of its ability to transmit in the human population. And so that's really complicated, and it's not possible to measure human transmissibility with high accuracy in the lab. So instead, in our experiments, we're simply focusing on measuring the biochemical phenotypes that we think most crucially contribute to fitness. And for the receptor binding domain, we think these are the following phenotypes. This receptor binding domain has to fold properly. Once it's folded properly, it has to bind to ACE2 with high affinity. And then the virus will benefit if it can change its ability to be recognized by antiviral neutralizing antibodies. So at sort of an approximate level, we like to imagine the evolution of this protein occurring under the following biochemical constraints. There's evolutionary pressure to maintain these two phenotypes of RBD folding and ACE2 affinity, while changing this phenotype of recognition by antiviral antibodies as much as possible. And so to measure these biochemical phenotypes in high throughput, we've been primarily using this really simple yeast display system. So we take advantage of the fact that because the RBD is an autonomously folding protein subdomain, we just express it on the surface of yeast. And then we can easily measure how much RBD is on the surface of any given yeast using an epitope tag. And we can also incubate the yeast with secondary molecules such as ACE2 or anti-RBD antibodies to see how much binding there is of the RBD to those other molecules. And so importantly, this basically means that the yeast sort of gain a fluorescent signal proportional to these different biochemical properties. So for instance, if you titrate in the concentration of ACE2 at different levels and then run the yeast through a flow cytometer, you can actually measure biochemical properties just like you could in a conventional biochemical assay. But your readout can entirely be on a flow cytometer. And so the advantage of transferring the reading to a flow cytometer is not there if all you're looking at is a single variant of the RBD. However, the advantage comes if you want to look at a lot of different mutants of the RBD, because we can now make a large library of yeast, each of which have a different RBD mutant on their surface. And then the yeast are actually engineered to have a 16 nucleotide barcode, which tells us which RBD mutant they have on their surface. So we can now take a very large library of different RBD mutants, thousands or tens of thousands or even a couple 100,000, and run them through a flow cytometer and separate them based on their biochemical properties, and then just sequence this barcode to sort of determine the biochemical properties of these very large numbers of mutants, all in a single experiment. And so this gives us data like what I'm showing here. So hopefully you can see a heat map. If you're not seeing a heat map, someone please interrupt me and let me know. So these heat maps are showing how mutations affect two of the biochemical properties of the receptor binding domain. The first property is ACE2 binding. This is the change in log 10 dissociation constant for dimeric ACE2. And then here's expression of RBD on the surface of the yeast. And then in these heat maps, each cell represents a different mutation. So like this is a mutation F347D. And the cells are colored so that mutations that are bad for that property, like this one, are colored red. Mutations that have no effect on the property, like this one, are colored white. And mutations that enhance that biochemical property, in this case of the top heat map, ACE2 affinity, are colored blue. So if you just sort of scroll through these heat maps, you see there's a lot of yellow because there's a lot of mutations, or sorry, a lot of white because there's a lot of mutations that don't have much effect on this biochemical property of ACE2 affinity. There's a lot of red because there's a number of locations in the receptor binding domain that if you mutate them, you basically break ACE2 binding. And importantly, you can look down here to figure out if you're breaking it by messing up the folding, which is sometimes the case, or breaking it by just messing up the ACE2 binding itself. And then there's occasional pockets of blue, such as this mutation here, N501Y, which enhances ACE2 affinity. And so these maps of the biochemical effects of these mutations are super useful if you're sort of monitoring the virus's evolution. So for instance, in late 2020, people started to note the rapid rise of lots of SARS-CoV-2 variants that had a particular mutation, N501Y. So we could immediately go look up that mutation and see, well, it increases affinity for ACE2, and that might have been part of the reason it was being selected. And in general, we can go and we can sort of look at any arbitrary mutation that appears in a SARS-CoV-2 variant of concern and see what is its effect on these biochemical properties. So this is very useful for sort of trying to interpret all of the genetic information which is coming out all the time. You can also sort of use this information to look at a lot about sequence structure relationships of the SARS-CoV-2 receptor binding domain, and some of that information can be visualized at this link here, but in the interest of time, I'm not going to go into that in this talk. Instead, I want to spend the last biggest chunk of the talk focusing on the question which I think is of most intense interest to people right now, which is how do mutations affect the ability of antibodies to recognize the receptor binding domain? And so, as you can imagine, it's very simple to use this yeast system to measure that. We can just incubate the yeast with a fluorescent antibody and sort, for instance, for yeast that have receptor binding domains that escape binding by the antibody. And when we do that, we get data like these. So first I'm showing you what escape looks like from a single monoclonal antibody. And so this antibody is called LY-CoV-555, and it's an antibody that was developed as a monoclonal antibody drug by Eli Lilly under the name bamlanivimab. And so here I'm showing you some information about this antibody and the receptor binding domain. So first, here's the crystal structure determined by Abcelera and Eli Lilly of the antibody in pink bound to the receptor binding domain, which is in gray. And so what we did, which was new, they'd already solved this crystal structure, is we used this yeast display system to go through and for every single site in the receptor binding domain, determine how all of the mutations at that site affected binding by the antibody. And so sort of the average effective mutations at the site are shown here on this plot. So you can see at most sites in the receptor binding domain, mutations have no effect on binding by this antibody. But there's a few sites like 452 right here, 484 right here, and a collection of other sites right here where mutations strongly reduce binding by this antibody. And importantly, you can see all of these mutations are in the physical interface of the antibody and the receptor binding domain, although not every mutation in this physical interface is an escape mutation. This is sort of a common thing that's seen as these hotspots of escape. And so this information turned out to be really useful to know because today, essentially every SARS-CoV-2 variant out there has mutation either at site 452, which is mutated in the Delta variant, or 484, which is mutated in like the mu and the gamma and the beta variants. And the sort of result of this is that almost as soon as this antibody received emergency use authorization by the FDA, it became obsolete due to first the widespread appearance of this mutation and then the widespread appearance of this mutation. And for that reason, this antibody is no longer being distributed as a treatment for SARS-CoV-2. So one of the things you can imagine is that if you were going to kind of do this all again and spend however much money Eli Lilly spent to get emergency use authorization for this antibody, which is probably quite a bit, you might want to figure out what these escape mutations were ahead of time. And then you could try to choose an antibody that you thought was unlikely to have escape mutations, both by looking at what's out there in nature and also using data like what I described earlier in the talk that shows you how tolerant each site of the receptor binding domain is to mutations. And so to sort of help with those types of efforts, we've repeated the experiment I just showed you for lots of antibodies. So here's a map that's just sort of summarizing the results from lots of antibodies. So right here are the set of antibodies that we've characterized so far. These are primarily antibodies that we characterized in collaboration with the Crow lab, with Vir Biotechnology, or with Michelle Nussenswag's lab from Rockefeller. And so what I'm going to do here is show it like this. So if we click on any given antibody, so here's the one I was just describing, you can see its escape profile sort of highlighted here and then shown in isolation down here. So I was just telling you about LY-CoV-555 or bamlanivimab, which is escaped by mutations at sites like 452 and 484. And so that, as we know, turns out not to be a very good antibody to use. because it's escaped by these common mutations. And if we sort of organize the antibodies in the space of escape mutations, you can see that LY-CoV-55 is sort of a very typical antibody that's very similar to a lot of other antibodies, like several isolated by the Rockefeller group, some isolated by the Crow lab, some isolated by Vir. So it has sort of escape mutations that are quite common. However, there are other antibodies like S309, which sits over here, or S2H97, which sits over here, which have very distinct sets of escape mutations. So for instance, S309, which is an antibody that's been developed for human use by Vir Biotechnology, is only strongly escaped by mutations at sites like 337 and 340, both of which are relatively mutationally intolerant sites where variation is not appreciably seen in SARS-CoV-2. Similarly for S2H97. And so we think a benefit of having this type of information is that for the development of monoclonal antibody therapeutics, you can sort of take any given antibody, see both how kind of unique it is, see where there are going to be escape mutations, and then compare that both to the type of data on functional constraints I was describing and natural evolution to maybe try to choose antibodies that are unlikely to have escape mutations. And then also for whatever antibodies that have been developed, you can kind of easily track when there might be mutations that could start to cause problems. And so these data are all sort of available and will hopefully help promote that goal. However, that's all for monoclonal antibodies. And although it's certainly useful to have monoclonal antibodies in the sort of toolkit for treatment of severe COVID-19, as I think you all know, it's really polyclonal antibodies that we're counting on are sort of already getting us out of the pandemic. And so just to make the distinction crystal clear, a monoclonal antibody is a defined chemical compound that's usually produced recombinantly nowadays that binds to a specific part of a virus. And so typically, if you get a mutation where the monoclonal antibody binds, it doesn't bind anymore, and the virus is resistant, just like I described for LY-CoV-555. However, one of the great things about the human immune system is that the human immune system in response to vaccination or viral infection makes a polyclonal antibody response. And the polyclonal antibody response potentially consists of lots of different monoclonal antibodies, each of which can potentially bind to a slightly different part of the virus. And so this polyclonality of the human immune response can make it much more resistant to viral escape. So for instance, if this virus gets a mutation that knocks out the purple antibody, you still got the gray and the blue and the green one left. However, something I want to emphasize is that the sort of breadth or functional polyclonality of the antibody response is sort of in principle. There's no fundamental law of immunology that says that the immune system can't happen to make a whole bunch of antibodies that pretty much bind to the same part of the virus. And in fact, we know from comparative studies of other viruses that the extent of focusing of polyclonal antibodies is a key factor in shaping viral antigenic evolution. So for influenza virus, for instance, Juhei Li in my lab showed a few years ago that the human polyclonal antibody response is often highly focused, at least the neutralizing component of it is often highly focused, such that it's not uncommon to be able to find single mutations to flu hemagglutinin that reduce neutralization by human polyclonal serum antibodies by greater than 10-fold. And the fact that the virus can often get these huge jumps in polyclonal antibody escape just with single mutations is probably one of the reasons that influenza is so adept at evolving to erode human antibody immunity. In contrast, for the measles virus, a group at the Mayo Clinic recently put out this very nice paper showing that the polyclonal antibodies in the sera target many epitopes with similar potency, such that no single mutation has a large effect. And this may very well be one of the reasons why measles is pretty ineffective at evolving to erode the polyclonal antibody response. So what is the focusing of the antibody response to SARS-CoV-2? So first, if we just look at the domain level of the spike, we find that the neutralizing antibody response is not only focused on the spike, it's heavily focused on just the receptor binding domain, which is really only, less than 20%, about 15% of the total spike protein. And to measure this, what we did is we took human convalescent sera, so these are people who recovered from SARS-CoV-2 infections, and we measured the neutralizing titer, so shown here on a log scale on the y-axis. And so as I described, these neutralizing titers range from about 100 to as high as 10,000. And then what we did is we just biochemically depleted from these sera just the small fraction of antibodies in the sera that specifically targeted the receptor binding domain. And then we measured the neutralizing titer again. And so you can see these lines slope down, often by a lot. And recall this is a log scale. So what in effect this is showing is that in virtually every case, depleting the RBD binding antibodies reduces the neutralization titer by, at least twofold and often by more than tenfold. So in many cases, more than 90% of all of the neutralizing activity is just due to antibodies that are binding the receptor binding domain. This is true in other collections of sera as well. So here's convalescent sera collected a couple of months after infection. The same thing is true if you look at convalescent sera from later time points, and is actually even more true if you look at sera from the Moderna mRNA 1273 vaccine. So again, this neutralizing activity of the polyclonal antibody response is quite focused on the receptor binding domain. And when we look within the receptor binding domain, so using that system I described to you to map where in the receptor binding domain mutations escape neutralization, we find that in many cases, particularly for infection, less so for the mRNA vaccine, a single sort of functional class of antibodies makes the largest contribution. So to sort of explain this, here's this display that I was showing earlier, where I've shown a bunch of monoclonal antibodies, and they're sort of grouped in these different colored classes based on how similar their escape mutations are. So if we now add to this plot convalescent sera, and we're now just asking where in the receptor binding domain do mutations reduce binding by convalescent sera, those are these dark green points here. And you can see that many of these points sit right over here with the class 2 antibodies. So very similar escape mutations, just this single antibody class. And in fact, if we just look at the convalescent sera, we can see there are sites where mutations, particularly sites like 484, where mutations can have a really huge effect on binding, and it turns out also neutralization by convalescent sera. So in many cases, it's quite focused with this single antibody class. Something that I think is a little bit optimistic is that when you look at serum antibodies elicited by the Moderna mRNA 1273 vaccine, they sit a little more centrally in this cloud of antibodies. And so for some reason, in many cases, the mRNA vaccine is inducing sort of a more balanced set of antibody specificities within the receptor binding domain, which could turn out to be a good thing. Okay. And then another thing that we're sort of thinking about going forward in the lab, and this is one of our most intense areas of interest right now, is, all right, if these polyclonal antibodies can bind multiple epitopes, what will viral escape over the longer term, like several years, look like? And so again, you can break down the epitopes in the receptor binding domain into different sort of approximate classes. We're using a classification scheme developed by Christopher Barnes and Pamela Bjorkman. And in this scheme, we call the potently neutralizing antibodies class 1, class 2, or class 3. And as I mentioned, class 2 tend to be immunodominant in the response to infection, at least with early SARS-CoV-2 strains. And the idea of these epitope classes is that most antibodies pretty much bind, or at least most neutralizing antibodies pretty much bind in this blue region, this purple region, or this pink region. And often a mutation that escapes antibodies, one antibody of a class will also escape other antibodies of the class. So you can then think that really full erosion or large-scale erosion of the antibody response is going to require the virus to get mutations in each of these classes. And so again, we can go back and we can try to ask the question, sort of what would escape from each of these antibody classes look like? And I'm going to unselect class 4 because it's not as potently neutralizing. So if we just look at class 2, which is the immunodominant ones, we see that mutations at sites like 484 really strongly escape antibodies in that class. Also mutations at sites like 452, which is mutated in the Delta variant, have the same effect. And in fact, most SARS-CoV-2 variants, one of the things they have is a mutation in the class 1 site. After that class 2 mutation, you can say, what about the class 1 site? So antibodies that target the class 1 site tend to be escaped by different mutations. So they're not much affected by mutations at 452 or mutations at 484, but they are affected by mutations at sites like 417. And in fact, many SARS-CoV-2 variants of concern have picked up this mutation, K4179, after they've gotten their class 2 escape mutation. So sort of after they've gotten rid of the dominant antibody class, they're now escaping a second antibody class. And then there's this third antibody class, which is escaped especially by mutations at sites like 346, also at sites like 449, which is actually a site where mutations are coming up in a new SARS-CoV-2 variant that's been mentioned, and sort of other regions like that. Fortunately, so far, most major SARS-CoV-2 variants have not managed to combine escape mutations in all three of these classes. And then the last point I would just like to make is if you take all these monoclonal antibodies we've looked at, you can kind of see where are the escape mutations on average from all these antibodies the largest. And when you do that, you see the peaks are at positions like 490, 484, 452, 417, 346. And all of these are sites that have been recurrently mutated in different SARS-CoV-2 variants of concern. So I think this really shows the virus is picking up mutations at the sites where mutations have the biggest effect on antibody binding, and that's probably a major thing that's driving the virus's evolution. And so hopefully we can kind of keep on top of this and monitor it as the process continues. Okay, so to just sort of conclude, I just want to make the point that even though we're very focused on SARS-CoV-2 right now, really relating changes in genotype to phenotype is this old challenge in evolutionary biology. People were worrying about it before 2020. Zucker, Candle, and Pauling were worrying about it in 1965. And obviously, we're still worrying about it in 2020 and 2021. And I would, in fact, argue that if you're interested in studying viral evolution, this question of interpreting the phenotypic effects of mutations is increasingly becoming the bottleneck, because people have gotten so good at sequencing these viruses that obtaining genotypes, which also used to be a very difficult thing, is getting easier and easier and easier. And so I've kind of described to you some of the ways that we're working on scaling up the characterization of phenotypic effects of mutations to a scale that's at least quasi-comparable to the scale of genome sequencing, viral genome sequencing, so that hopefully this interpretation can be done better and faster. And finally, I'd like to end with some thanks. In particular, I'd like to thank Tyler Starr, who's this really brilliant postdoc in my lab who is developing this entire yeast display system for some other purpose and only switched to working on SARS-CoV-2 in early 2020, and then has basically led and developed a large fraction of the work I described. And Ali Greeny, who's a really excellent MD PhD student in my group, who was also working on a different project prior to 2020, and then started working with Tyler on the yeast display system and has subsequently extended it to a lot of these really cool questions about SARS-CoV-2 antibodies. I'd also like to thank Rachel, who did the work with coronavirus 229E, Sarah, who developed a lot of the computational visualizations, our interactive visualizations, and Kate and Andrea, who did a lot of work on sort of neutralization assays that validated many aspects of these results, even though I didn't show a lot of that validation today. And finally, I'd like to thank our collaborators and my funding sources. The slides are available here, and hopefully the link is in the chat, and I'd be happy to answer any questions.
All right, really great stuff, Jesse. Great job. So we have several questions coming into the chat Q&A. So first one I'll ask on Mehdi's behalf. You mentioned optimizing antibodies for SARS-2 neutralization. Is there a specific and coherent part of spike that could be targeted using a monoclonal antibody, in your opinion, or do you believe that monoclonal antibody therapies are doomed to fail?
So I certainly think that monoclonal antibody therapeutics are going to be a really useful thing. I mean, there's plenty out there that the virus has not escaped from. I mean, whether there will be a monoclonal antibody that's developed that works for the next 50 years, I don't know, I wouldn't count on it. But I think there's certainly going to be possible to develop monoclonal antibodies that will work for quite a while. And in fact, I would argue that maybe the best approach, although obviously you want to choose as good of antibodies as possible, maybe the best approach is not only to look for sort of the silver bullet monoclonal antibody, but just develop several different monoclonal antibodies and then use information like what I was presenting to just make sure you're choosing an antibody that is not escaped by the viral variants that are present in that neighborhood. So I certainly think it's a promising approach. Although, I mean, obviously, there's a lot of reasons why vaccines long-term are, more scalable and better as a real long-term solution.
Right.
So the next question I see is, do antibodies have to block the RBD to be effective neutralizers? No, they do not. So there are neutralizing antibodies against the N-terminal domain of SARS-CoV-2, and there have been a few described against the S2 subdomain. And I should mention, although I didn't go into it in detail in this talk, exactly how important the RBD is versus the other epitopes depends in part on the specific system that's being used to measure neutralization. So far they all sort of describe RBD antibodies as being the dominant component of the neutralizing antibody response, but some systems measure them as like much more important than they say the NTD antibodies, and others kind of put them as moderately more important than the NTD antibodies for overall neutralization. That said, within the polyclonal response for any given person, there can be antibodies targeting a variety of different epitopes. But so far, the natural immune response and the vaccine-elicited immune response, RBD binding antibodies are the biggest component of neutralization.
And I guess I could help you here, Jesse. Frank asked is something about measles here. Is there less selective pressure for measles to vary surface antigen since the virus often infects and kills memory B cells?
Possibly, it could be something like that. I think this is, although I personally doubt that's the case. So why measles doesn't undergo antigenic evolution is sort of like a difficult question. It could be that somehow mutations that escape antibodies in the measles protein just somehow break the protein in a way that decreases viral transmissibility. It could be that the human immune response or antibody response to measles is much better balanced, as I was describing. This is sort of the question that's like really hard to answer experimentally. I mean, certainly you can select escape from monoclonal antibodies to measles in the lab. So it's not like it's impossible for the virus to get a mutation that escapes the antibodies, but it doesn't happen in the human population. And, in the end, we don't really know. I mean, there's other RNA viruses like poliovirus, for instance, which also doesn't undergo much antigenic drift. Why? I don't know for sure. I mean, in poliovirus, I've always felt like maybe it's because it's a non-envelope virus and, the proteins are much more kind of packed in on the surface, and there's probably potentially a lot less room to change those proteins without messing something up. But I think it's an open question. I mean, some RNA viruses undergo rapid antigenic evolution, some don't. And like, why is a fascinating question.
So we have another question about from someone anonymous, characterizing mutations in other viral proteins, looking at host viral interactions using your approaches. Are you thinking about expanding outside of spike?
Yeah, so definitely. I mean, so we're trying to do a couple of things. One is that we're working on a system to look at the entirety of spike and not just the receptor binding domain. We've also done quite a bit of similar work with influenza virus, hemagglutinin. We're doing some similar work with HIV envelope, and then we're working on setting up sort of a generalized deep mutational scanning system that would hopefully work for any or at least most class 1 and class 3 viral fusion proteins. So we're very interested in doing that. So far, most of our work has been focused on sort of the entry proteins of the virus, like the fusion or receptor binding proteins. But certainly, I think similar approaches could also be applied to, other viral proteins. The main thing is you just need some sort of effective high-throughput selection scheme.
Mutations.
And we like collaborating with people on this because we develop all these computational and technical tools. So if you have ideas, let us know.
Next question, mutations reduce neutralization of the antibodies. However, is the remaining neutralization enough to have a protecting effect in vivo?
Yeah, I mean, this is a great question, right? I mean, the question comes down to like, what is the relationship between in vitro neutralization and protection? And it's not known. I mean, there is some evidence at this point that neutralizing antibody titer is a correlate of protection. So there's some quantitative relationship between neutralizing antibody titer and protection, but like exactly what that is not known. Another important question is, even if you're able to be reinfected at some reduced neutralizing antibody titer, how sick do you get? I think these are really important questions for figuring out sort of how dire will be the public health consequences of the viral antigenic evolution. I certainly think that because of viral antigenic evolution, if people aren't repeatedly getting updated boosters, at some frequency, which is not entirely clear yet, people are going to get reinfected. They're probably going to get reinfected regardless. I mean, that's what happens with common cold coronaviruses. How sick they're going to get and how often they get reinfected are going to depend on questions like the ones you're asking, which I think in the end will only be able to be answered by sort of epidemiological studies in humans.
And Ujjal's got a great question here. He's just talking about antibody treatments. Now that antiretroviral drugs are coming online from Pfizer and Merck, what's your thought now about using antibodies as treatment? And can you think of specific situations where they'd be particularly pertinent, I guess is what Ujjal's asking.
I think that's a great question. I'm too far away from like the clinical end of the use of these to really comment on what would be the advantages of drugs versus monoclonal antibodies in different situations. I mean, I would say since it's an evolving virus, probably having several of these things in the arsenal would be good. And for all of them, the main problem is going to be, as is the case for other respiratory viruses like influenza, that these things are most useful if they're given early on. And the problem with drugs against influenza is often that by the time someone gets the drug, their viral titers have already peaked and they're sick from something other than acute viral replication. But I certainly think both the drugs and monoclonal antibodies are probably going to have a role going forward. But I think someone with more clinical expertise would probably be able to say what they think are going to be the pros and cons of them.
And have you looked at this new, I guess, spike mutation in the Delta Plus? Have you gone? I guess is there one mutation or a couple mutations in spike?
Yeah, there's a lot of Delta Plus variants. I actually can't quite remember which one is in the UK, the one that was described a lot in the UK, but certainly there are some Delta variants that have K417N, which is a mutation that reduces binding by some antibody classes. There's some that have E484Q, which is a mutation that strongly reduces binding by other antibody classes. And I do think that probably what we're going to see is the next, I think there are always going to be sort of next variants of SARS-CoV-2 that have mutations that further reduce antibody recognition, and probably the next one is going to come out of Delta. And so probably one of these Delta Plus variants, but I don't think it's clear at this point whether any given Delta Plus variant has like a really clear edge over others at a global level.
Let me just, I can ask one quick question. You showed that great analysis on 229E. Have you, or actually can you do this type of analysis with these other common cold coronaviruses, Jesse?
Yeah, we've been trying. We actually wanted to do it with OC43 originally because 229E is actually an alpha coronavirus and OC43 is a beta coronavirus. I think there's something with OC43 where maybe the receptor for the lab adapted OC43 isolate, which is the one that everyone uses, which was, passaged in, like a lot of these older things passaged in this whole host of cell and animal systems for a long time, might be slightly different than what the actual current human isolates are using, because we can pseudotype lentiviral particles with the lab adapted ATCC OC43 spike. We have been unable to pseudotype it with any of the human isolate spikes we've tried, so we aren't really able to do the experiment, and that's got to be due to some change in receptor usage between the two, we think.
Maybe I can ask a question also, Jesse. Great talk, thanks. So like looking into the future, is there anything from your analysis in terms of mutation that the current variants haven't exploited, but is on your list as something that would be detrimental in terms of, antibody evasion in the future?
Yeah, I mean, there's certainly, I mean, the problem, and this is why I always kind of try to describe our goal of our work as interpreting rather than predicting the virus's evolution. There's actually there's not like an infinite number, but there's like quite a few mutations that could mediate antibody escape that the virus hasn't gotten yet. And so, although I think we can sort of say the future variants will probably have mutations at some number of the subset of about 20 sites, I think it's very hard to know like exactly which mutations they will have. That said, I certainly think that probably mutations in like this region from 443 to 450, which is really importantly targeted by these class 3 antibodies. I think that's one region where there's a lot of room for future antigenic escape. And also, Delta doesn't all have a 484 mutation. There could well be one there. And then I think it will also get increasingly complicated because as the virus continues to evolve, exactly which mutations are tolerated are going to change. And so, for instance, we've also done some work where we've looked at the effects of mutations in the backgrounds of several different RBD variants. And in some key regions, you can see that just a few mutations shift to some extent the mutations that are tolerated at other sites. So yeah, I think we can kind of like narrow down what are going to be the sites of important mutations well enough that when they pop up, we can say that's likely to be an important mutation. But I don't know if we're going to be able to predict what they're going to be. We're going to have to more like diagnose it very quickly after it happens. So.
We got one more question here before we wrap up. Could other antibodies that do not neutralize the virus still have a protective effect by acting in other immune pathways?
Yeah, that's a great question. I mean, so, and To be correct, I was talking about how neutralizing antibodies are a correlate of protection. Formally speaking, on the studies that have been done so far, neutralizing antibodies and spike binding antibodies and RBD binding antibodies are all about equivalent correlates of protection. That said, I think that probably, I think that's partly because right now, the way that the immunization and viral evolution has worked, that those things are all highly correlated with each other. Like people who have high spike binding antibodies tend to have high neutralizing antibodies. That said, I still think that the neutralizing antibodies are likely to be the most important mechanism of protection. This is just an opinion, but the opinion is based on the fact that when you look at something like coronavirus 229E, antibodies are binding through lots of parts of the spike, but the neutralization or the mutations that are actually occurring are at the sites where there's the biggest effect on neutralization, like in the RBD receptor binding loops and to some degree the N-terminal domain. That said, I don't think that means that non-neutralizing antibodies don't do anything. It just means they're not putting as strong of pressure on the virus. They could be, providing less potent protection. They could be ameliorating severity of disease, things like that. I think more studies will be needed to look at that. And then certainly, there are other mechanisms of immunity like CD8 T cells, which are not going to prevent you from getting infected, but could take a chunk out of the severity of the disease if you do get infected.
All right, we're down to our last 30 seconds here. So great talk, great turnout, great questions. Thank you for sharing your exciting work, Jesse, and we owe you a dinner here in San Francisco soon, I hope.
Okay, thank you, Nevan. Bye.
Thanks, guys.